Hello, everyone. Welcome to today's webinars hosted by Medican Express. Thank you for joining us. Today's topic is on innovative drug delivering techniques for researchers with neurocell systems, which addresses many of the long-standing challenges in neurobiology. Today, we are very pleased to have Dr. Chuan Chao, Associate Professors in Taipei Medical University, as our guest speaker. Dr. Chao is a distinguished interdisciplinary biologist. His research spans from biochemistry, genetics, and nanotechnology. He earned his PhD from Academia Sinica. Then he was trained extensively in different research groups in Kyoto University before he started his independent laboratories at Taipei Medical University. He has over 2 decades of research experience and prestigious award, such as the AMBO Global Investigators Award and speaking invitations from leading institutes like EMBL and Academia Sinica. Dr. Chao is currently leading a pioneering works on genomics dynamics in polyploid tissues and innovative molecular delivery systems. He's going to show us today. So without further ado, please join me in welcoming Dr. Chuan Chao.
Okay, thank you. Thank you all for attending this talk. First, I need to thank Medican Express for the invitation, and thanks Dr. Lance for introduction. I'm from Taipei Medical University, Taiwan. It's my honor to be here and share my study with you. So today I will show you how to use this very cheap homemade reagent to deliver exogenous material into primary neuron and neuron stem cell for the cellular and the molecular biology investigations. Okay, for a long time, we often mark our progress in science by improvement of image. So as you can see, this first Nobel Prize, the physical prize of 91st, was an image prize given to the Will Longin for his discovery of X-ray and the astonishing ability to allow the non-invasive detection of internal structure within the human body. So this allows doctor to visualize the bone and the other internal tissue without surgery by using this pioneering radiation imaging technology. A few years later, in 1996, the Nobel Prize in Physiology and Medicine was awarded to the Camille Gauge for his development of a silver nitrate staining to visualize the morphology of nerve cell in the brain. Over the last century,
I don't think. Sorry, excuse me. I don't think it's not shared the screen. On my,
really?
I can't see. Yeah.
Okay, so so let me try. Share this screen. Can you see this one?
Yes, has started. Yes.
Okay, so do I need to start again? Okay.
Yeah, yes, please. Yes, I don't think it's shared. Yeah.
Okay. Sorry. So over the century, these numerous advanced imaging technology have been honored with the Nobel Prize to highlight their deeply impact on science and medicine. So this technology, including the X-ray crystallograph, electron microscope, phase contrast microscope, MMR, MRI, and CT scanning, among others. So they have dramatically changed how we see everything from the tiniest atom and the cellular structure to the vast cosmic system. So this breaking through continue to shape how we can explore, understand, and innovate across the discipline. So as the saying goes, this seeing is believing. So many great scientific discovery begin with simple observation. So the more we can see, the more we can explore. For the centuries, people have been attracted by the animal that can grow by themselves, showing off the beautiful and the shifting colors. Since the moment, people wonder how can they do that. So it's stayed a mystery for a long time until 3 Nobel Prize winners finally cracked this code. In 2008, Osamu Shimomura, Martin Kelfi, and Roger Chen won the Nobel Prize in chemistry because of their study on bioluminescence. For this, Dr. Shimomura, the first people discovered GFP from jellyfish and demonstrate its unique ability to emit green fluorescence under UV light. And for the Dr. Kelfi, he demonstrated that GFP could be used as a fluorescent marker in a living cell without need for this additional cofactor, making it as a powerful tool for studying gene expression, protein distribution, and the cellular process. For the Dr. Roger Chen, he made a revolutionary contribution in fluorescent protein engineering for the development of a multi-color variant of this GFP, which expands its image possibility to visualize and track cellular process by multiple colors simultaneously. So because of their study, today we not only understand the mechanism of bioluminescence, but also turn this glowing magic into the most powerful tool which can detect fluorescent label protein in living organisms for gene expression tracking and living cell image by watching. So now we have such a powerful tool to let us to see the intracellular and the molecular interaction directly. But it still have a big challenge to deliver this reporter gene into the cells efficiently, especially under the primary culture system. So as you can see, this image shows two different neurons labeled with transfected GAP and M-cherry respectively to outline their morphology. Based on the fluorescent signal, we can obtain the detailed morphology change of the neuron under either culture or in vivo system. So this is the first time that we can deliver various fluorescent genes into different primary neurons in the same culture well sequentially, and then monitor the interacting of a presynapse and the postsynaptic structure under such a high resolution state. So today I will show you how to use this very cheap homemade reagent and the two simple step method to deliver exogenous material into primary neuron and the stem cell for these cellular and molecular biology investigations. So today, numerous transfection strategies based on the physics, chemical, and the biological approach have been developed to accommodate various experimental conditions. For the physical method, it can deliver a nucleic acid directly into the cytoplasma or nucleus through this high-intensity physical force like electric pulse, gene gun, microinjection or mechanical stress to increase the permeability of a cell membrane. However, this physical force can also cause severe damage of a cell membrane and high mortality after treatment. And moreover, this high technique skill and the expensive equipment are always required for this physical gene delivery method, which is unfriendly for many labs. For a biological method, it always have a great gene delivery efficiency, but this complex gene construction and the higher biosafety level are still the major concern for many labs. So today, this chemical method still the dominant strategy to deliver various exogenous material into cell. It uses the carrier molecule that can neutralize or impart a positive charge onto the nucleic acid. The genetic material from these complexes with this reagent molecule for delivery, which protect them from this degradation and facilitating the cell entrance. Actually, this chemical transfection generally fall into the three categories, including this lipid-based polymer and the calcium phosphate technique. For the lipid-based transfection, liposome or cationic lipid is employed to form these complexes with the genetic material. And the liposome method, like lipofectomy, usually encapsulate this nucleic acid within the aqueous compartment or a bilayer of a liposome for the delivery. So this complexity can be uptake directly by the fusion with the cell membrane to release the DNA construct inside the cells. For the cationic polymer, they are a macromolecule with a multiple positively charged group along this polymer backbone. And this molecule form the different complex with nucleotide when this electrostatic interaction. And this complex will be typically uptake by this endocytosis and then released into the cytosol through the endosome escaping mechanism. The cationic polymers are convenience tool for this gene delivery, but more cytotoxicity because of this strong electrostatic interaction that can disrupt these cellular membranes. Actually, this calcium phosphate precipitation has been a popular transfection method since its introduction in 1973. Because it requires only easily available and inexpensive component. The principle of calcium phosphate delivery involved in this mixing DNA with calcium chloride in Hippies buffered saline phosphate solution to generate a calcium phosphate DNA co-precipitate. So in this co-precipitate, the calcium phosphate facilitates binding of the condensed DNA to the cell surface by eliminating the negative charge of the nucleotide and then promote the DNA entering into the cell by endocytosis. So this table shows you the comparison among these three different methods. For the physical method, they are useful for the super difficult transfected cell, but may cause this damage and usually lower tissue specificity. And for this biological method, such as a virus, offer high efficiency and long-term expression, but the high risk of biosafety, immunogenicity, and genomic integration are still the concern. And for this chemical method are relatively safe and scalable, but may suffer from this low efficiency and transient expression. Actually, this optimal method depends on this experimental design and objective. But in general, chemical method is this optimal choice for most of experiments. So today, this calcium phosphate method still a popular transfection strategy because it's require very cheaper and more convenient component than other chemical method. So as you can see, this table shows the summary of a conventional method for the calcium phosphate-based transfection. Over 30 years, many calcium phosphate-based transfection tools have been developed, but the formation of an optimal calcium phosphate nanoparticle is difficult to reproduce. So it's make this protocol can only be applied in some specific condition and criteria, which constrain this applicability. So here you can see the final calcium concentration they use about 250 millimole, which is an extremely high concentration that can cause the huge calcium phosphate precipitate, but not this nanoparticle in our system. And recently, the formation of crystalline calcium phosphate has been gained attention because of its applicability in biomedical material for clinical therapy. And importantly, recent study has shown this formation of calcium phosphate precipitate does not follow the classical growth mechanism for the crystal formation. They found the small pre-nucleation cluster (PNC) are formed initially and then aggregate into the branch polymeric assembly. So this is followed by dissolution and the localized reprecipitation process, which give rise to the spherical domain and then to facilitate the formation of amorphous calcium phosphate nanoparticle (ACP). Okay, so this amorphous calcium phosphate ACP show a size in 1 to 5 nanometer in diameter at the early stage, then transform into the 20 to 100 nanometer in size eventually. So which played a significant role in biomedical and material science application because of its high solubility and biocompatibility. So unlike other chemical transfection reagents such as cationic polymer or liposome, calcium phosphate nanoparticles show minimal cytotoxicity to make it suitable for in vivo and in vitro applications. Okay, so this table shows the current method for the calcium phosphate nanoparticle production. But many limitations such as the high cost, complicated protocol, and the spatial equipment necessary and this poor reproducibility remain a big issue. And multiple parameters show to involve in the formation and the solubility of calcium phosphate nanoparticle. And this appropriate window of condition for nanoparticle formation seems to be narrow. So this causes this major challenge of the low transfection efficiency in this primary culture cell. And the recent study has demonstrated that the formation of a calcium phosphate nanoparticle can be kinetically controlled by the calcium to phosphate molar ratio. So we try to identify the critical point of this calcium to phosphate molar ratio for this calcium phosphate nanoparticle formation under primary culture systems. So based on the chemical characteristic of calcium phosphate, we identify a golden ratio between the calcium and the phosphate buffer that facilitate the formation of this calcium phosphate nanoparticle with less than 100 nanometer in diameter. So this is a perfect size for this cell uptake and extra low cytotoxicity. And we performed a systemic study and found that when the final calcium concentration below 35 millimole, the calcium precipitate can be reduced. The aggregation ability and express this nanoscale size particle. Compared to this 15 millimole, 35 millimole calcium chloride show a similar particle size with nanoscale, but express a higher particle number than this 15 millimole calcium chloride. So indicate that a 35 millimole calcium concentration play the most significant role in the first step to determine the size of calcium phosphate precipitate, and with a higher production of this nanoparticle number within this window of concentration. So to address the impact of a different medium system on calcium phosphate nanoparticle formation, several common use laboratory medium were investigated in our system. So we found that a neuron basal medium not only show a perfect property to generate this smallest size, but also provide this highest number of the calcium phosphate nanoparticle compared to other media. So for this nanoparticle elimination, we found this HbACS without calcium serve as the best buffer system to remove calcium phosphate nanoparticle after transfection. So this data indicate this calcium phosphate nanoparticle can be generated conveniently and simply by this using a spatial medium and within a unique calcium to phosphate ratio, which can be then removed quickly by this calcium-free HbSS buffer. So we then conduct the primary neuron culture to reveal the transfection efficiency under different calcium concentration in the median system. So the result indicate that a 35 millimole calcium concentration combined with the neuron basal median system can obtain this best transfection efficiency. If we focus on this MAP2 signal, this is a kind of a neuron marker. It shows that a calcium concentration below 55 millimole and using this neuron basal median can prevent primary neuron from cytotoxicity during this gene delivery. And we also found this transfection efficiency also show a positive correlation with the nanoparticle number and this incubation time significantly. Indicate this user can manipulate the transfection efficiency depends on these parameters and their experimental purpose. So we gave this strategy a new term named telefaction, which combine two words. Tele from the Greek and the faction is for this delivery, meaning this gene delivery method that is a time resolved and purposeful design for the goal-directed study. Okay, cell viability was also addressed by this Bristol Brew assay under different transfection strategy. And we can see this telefaction show this almost no cytotoxicity of a primary neuron under 15 and 35 millimole calcium concentration and this under this neuron basal median system. And moreover, this neuron marker MAP2 positive cell number was also consistent to this Bristol Brew cell viability assay. So telefaction caused this lower cytotoxicity and show no significant difference compared with this control group, which is this best gene delivery system than other methods. And actually, this downregulation of endocytosis in mature neuron, making it poor transfection efficiency after long-term incubation, especially after 2 weeks culture. So we sought to understand the transfection potency of telefaction on different culture day in vitro. So as you can see, this telefaction display the highest transfection efficiency at DIV7 and start to reduce at DIV11 gradually, but it still have 5 and 1.2% delivery ability at DIV15 and DIV30 respectively. And interesting, this number of glial cell was increased during this long-term culture, but glial cell always show lower transfection efficiency after telefaction, suggesting this telefaction is a good tool for neuron gene delivery, and as combined with this neuron-specific promoter. So we also compare the telefaction with current popular transfection kit. The data indicate that most commercialized kit causes severe cell death after transfection, showing a significant reduction of the neuron marker MAP2 signal. And for this Lipo 3000 and the Lipo Max, they show this lower cytotoxicity than other commercialized kit, but telefaction still express the best transfection ability than this commercialized kit. So this flow chart displays the overview of a calcium phosphate nanoparticle preparation and the process of telefaction. So in our system, this calcium phosphate nanoparticle were created by mixing this 2 mole calcium chloride solution with 2 times concentration of this HEBS through either this vortex or pipetting simply without any further incubation. So the conditioned neuron basal medium from primary neuron, this is another critical point to generate nanoparticle. So only this neuron basal medium can promote calcium phosphate nanoparticle formation. Non-neuron basal medium will cause enlarged calcium phosphate precipitate and sphere cell death. And after that, you will get this telefaction median, which included this conditioned neuron basal median and the calcium phosphate nanoparticle for the gene delivery. And you just need to replace this culture median with this telefaction median and incubate for the desired time, followed by a calcium-free HBSS wash to finish all the process. So the next question is, how can we use this telefaction for advanced scientific study? So here you can see this is a conventional method usually cause enlarged calcium phosphate precipitate and result in this poor transfection efficiency and the severe cell death. However, this telefaction method always obtain nanoscale calcium phosphate nanoparticle for higher transfection efficiency. So as you can see, this calcium phosphate nanoparticle show a vigorous Brownian motion. However, this conventional method usually create this enlarged calcium phosphate precipitate. Okay, and moreover, this telefaction can deliver this DNA, RNA, and the protein into the primary neuron and the stem cell, which can be conducted to several applications, including this neuron morphology examination, molecular and biochemistry analysis, high efficiency co-transfection, and neighboring neuron comparison, and this real-time monitoring of synaptic connection and the subcellular distribution of this fluorescent protein. So telefaction can provide a friendly use and a low-cost protocol without any spatial skill for this molecular and cellular biological study in primary neuron systems. So for instance, this telefaction can deliver a reported gene in primary neuron for neuron morphology examination. So as you can see, this is fine neuron architecture can be illustrated by the expression of GLP protein after telefaction, so which could be a powerful tool for examination of this fine structural change of a primary neuron. So as you can see, the cilia can be identified clearly by this magenta arrowhead, and this blue arrow indicate the axon terminus button, and this red arrow point out this M-pass button of the axon, and the green arrow indicate the axon growth cone. So we can also detect the fine structure of spine morphology on this dendritic shaft. So in addition, study of the molecule and the biochemistry change in primary neuron with genetic manipulation is a critical step to understand the mechanism of a target gene. However, this low DNA transfection efficiency remains a big issue today. So for instance, this reporter assay is an important approach to investigate this transcriptional and translational regulatory mechanism, but it still has a difficulty to deal with this primary neuron. So here we demonstrate that telefaction is a perfect method to overcome this long-term problem. So we found this telefaction can provide about median level of luminescence intensity from the firefly and the retina luciferase, showing this similar expression tendency like our previous study. And moreover, telefaction can deliver DNA and RNA for the Western blot analysis, showing this significant change of protein level under dose and the time-dependent manner. So these results indicate that telefaction is the ideal method to deliver nucleic acid into primary neuron for further molecular and biochemical study. And for some research purpose, such as co-immunoprecipitation and colocalization analysis, more than two plasmids will need to be delivered into the cell. So in this case, telefaction can provide high efficiency co-transfection of multiple plasmid for the exogenous gene co-expression in primary neuron. So as you can see, we mix both the GLP and the M-cherry expression plasmid for calcium phosphate nanoparticle generation, followed by this telefaction. And this GLP positive neuron always co-express M-cherry signal, showing this great capability of the telefaction for this multiple gene co-transfection. So another powerful function of telefaction is that it can deliver various exogenous genes into the primary neuron within the same culture well sequentially at different time points. So this allows user to monitor and compare the morphological change of a gene manipulated neuron side by side with a control cell in the same culture well. So according to this strategy, it's very interesting. We found that only a rare population of neuron can co-express multiple genes under sequential gene delivery, and around 90% of these transfected neuron can express only single delivered gene after telefaction. So under this strategy, we can compare neurons side by side with different gene expression. For instance, we deliver CMPR1 siRNA labeled by this M-cherry signal first to knock down this endogenous CMTR1 expression. And this is RNA capping methyltransferase for controlling this dendritic sheath outgrowth. And 2 days later, GLP plasmid was transfected by the telefaction sequentially, followed by this PFA fixation after 48 hours incubation. So as you can see, only this M-cherry positive neuron loss of endogenous CMTR1 signal, but not in adjacent cherry negative cell. So when we compare the neuron morphology side by side, you can see only this CMTR1 knockdown neuron show this abnormal upperization of a dendritic shift. So this sequential gene delivery protocol by telefaction can let us compare neurons side by side within the same culture well to prevent the bias caused by these different culture wells. So in addition, this real-time monitoring of dynamic spine-axon interaction is still a difficult challenge for the neurobiology field. And fortunately, based on this sequential gene delivery by telefaction, dynamic visualization in real time to record either heterotypic spine axon or axon-axon interaction is possible. Okay, so as you can see in this video, the plasticity between spine and the axon was recorded. Let's show this dynamic interaction for the synaptogenesis. So based on this method, we can conduct it for these various studies, such as drug or gene screening, which involved in this synaptogenesis and various cell-to-cell interaction. So delivery of exogenous DNA by this sequential telefaction provided this ideal system to investigate the morphological change of control and the adjacent gene target neuron. This can provide this artificial bias or prevent this artificial bias and caused by this different culture well. And actually, telefunction can also deliver protein into the primary neuron. Since this calcium phosphate nanoparticle has been demonstrated to form the complex with protein, named this calcium protein particle, CPP. So we wonder whether telefunction can generate a calcium protein nanoparticle for the protein delivery in primary neuron. So here you can see, in this case, the reporter gene can be transfected at div15 of a primary neuron, followed by this interesting protein delivery at div20. After 24 hours, the neuron will fix for the further immunostaining and examination. So in this case, cotectin binding protein 2, CTTNBP2, with GLP tag. This is autism spectrum disorder associated protein. It can binding to this F-actin and control this neuron morphogenesis. So it can be delivered into the cell by telefunction and show its biological activity by interacting with this actin stress fiber and the contractile ring. And this indicates telefunction is a new strategy to deliver exogenous protein into cell with its biological activity, which could be an approach to monitor this interaction and the subcellular redistribution of a target protein. So in the second case, this is a NUMA, a nuclear protein associated with mitotic apparatus. So this was conducted to investigate its nuclear distribution after telefunction. So you can see this NUMA GLP was successfully transported into this nuclei of this primary neuron after telefunction. So again, this demonstrates that telefunction could deliver exogenous protein into cell and still keep its biological activity after telefunction. So in addition, delivery exogenous gene into the neuron stem cell for study this molecule mechanism is a significant challenge, since telefunction has shown to efficiently deliver this nucleic acid and the protein in primary neuron. So we speculate that telefunction may have this similar capability in neuron stem cells. So as you can see, that transfection efficiency show a positive correlation with the calcium phosphate nanoparticle loading volume and got higher gradually when this nanoparticle number increase. And the telefunction also show this potential of the protein delivery into the neuron stem cell. So here you can see this cotectin binding protein 2 GLP was transfected into the neuron stem cell and show a colocalization pattern with actin stress fiber and express this normal biological function after telefunction in neuron stem cell. So in this primary neuron stem cell system, telefunction also show this great delivery ability of a nucleic acid and the protein, which could facilitate a broader comprehension of this molecule and the cellular analysis. Okay, so here we provide this detailed instruction and to enable user to get this high efficient transfection quickly, simply and cost efficiently. This can also enable to apply for the primary neuron and the neuron stem cell system reproducibly. So telefunction will be further modified and expand to the ex vivo and the in vivo application. So we wish this protocol could be a foundation for the development of a more advanced delivery strategies. Okay, so here we appreciate Taipei Medical University and NSTC for the funding support. And we also thanks AMBO for the special grant offering. And we are thankful to the Professor Yixun Huang for her generous support all the facility to this study. And I also want to thank all the people in my lab who work with me to complete this project. And finally, I was deeply appreciate our collaborator for from this board and in Taiwan for their kind support. Okay, thank you for your attention and pressure time.
Dr. Chap.
Yeah.
I can see that this techniques can be quite useful for lots of people in the labs because it's quite common techniques using the chemical way for introducing foreign genetic materials. So I myself have some questions. First, do you know how those nanoparticle crystals forms? Is it before you mixing the DNAs, the genetic materials, or is after you mixing those in a specific buffer they form those crystals?
Yeah.
I see someone's raising hands. Could you please type the questions in the question and answer box, and then so I can pass to the speakers. We have the first question from the audience. Nice presentations. Would telefactions work for large plasmids or long messenger RNAs? Is there an up size limit for efficient transfections?
Yes. So in our case, we try to use a different size of plasmid to test whether telefaction has its limitation, and we actually. So I have a slide show you here. So for your question, as you can see here, we have tried to deliver 3 different plasmid into the primary neuron at DIV 7 and the DIV 14, and we can get a good result. And that's all the plasmid can be delivered into the primary neuron with a perfect expression level. And so as demonstrated in this case, these 3 plasmid express GFP CMTR1, mCherry, and HASBF1 show this different subcellular localization in nucleus, whole cell, and cytosolic distribution respectively, and they have a different range of size from the AKB to this 12 base pair, so that we can successfully co-express in the same primary neuron following this telefaction. So actually, telefaction, I think it can deliver a huge size of a plasmid because it can deliver the protein. The protein always they also have a big size, so big molecule. So I think this telefaction can also deliver this high molecule like a protein or plasmid into the neuron. But we try the size from this 8 kb to 12 kb within this range. But above this range, we never tried before.
Right. Okay, great. So yeah, we have many questions actually coming in the chat box. So the next 2 questions all regarding the different cell types for these methods. Laura asked if it's possible to transfect the glial cells with these methods, and Zhixiang asked if is there any potential to apply this technique to like adherence and the suspension cell line?
Okay. Actually, we also try to use telefaction in different cell line. So in this case, how to say? Okay, so actually, telefaction is fundamentally based on this calcium phosphate co-precipitation. So it has long been used for transfecting various immortalized cell line. And in this figure, we use two human cell line, 293T and the HeLa cell to test this transfection efficiency of telefaction. And for the 293T cell, so it's as you know, it's a common used human embryonic kidney cell line, particularly for this gene expression and the viral production. So we can see this telefaction can obtain over 90% transfection efficiency under 2-hour incubation with this calcium phosphate nanoparticle. And for this HeLa cell, this is a human cervical cancer cell line. It shows about 70% transfection efficiency under 2-hour calcium phosphate nanoparticle incubation. And actually, we also try COS7 cell line. This is a Africa green monkey kidney cell that we can have about 60% transfection efficiency by telefaction. So this result show this cell line, they are more robust than primary neuron. That's telefaction protocol can be adapt to this cell line system with a high transfection efficiency, at least in this 293T and the HeLa and the COS7 cell lines.
Right. Okay. We have many more question comments. Anthony asked, he's wondering if whether the nanocalcium particles can mediate the delivering this delivery system is 100% reproducible, and how long you can keep these forming the nanoparticles.
Okay, so I also prepared this table. So okay, so not this one. Show you another one. So how long this reagent can be stored, right? Okay.
Yeah.
Okay. So actually we try to test how long for this telefaction reagent can be stored. So actually this pH values stability of this 2xHEBS is a very critical issue to produce this calcium phosphate nanoparticle. So we try to understand how stable is about this 2xHEBS under different conditions. So as you can see, this up left table, we demonstrate that this 2xHEBS is super stable under this harsh condition. That all can give a good gene delivery into the primary neuron. And the below table show this storage condition and the stability during duration of the telefaction reagent and the buffer. So as you can see, exception of the telefaction master mix should be prepared and used freshly. And for the other telefaction reagent and the buffer, they are very stable and can be stored under regular condition for a long time. Yeah.
Okay. Good. Anthony also asked, does the size of the protein affect the telefaction's efficiency?
Oh, yes, we try different size protein, including just show you before NUMA and the CTT binding protein too. And then we also try this antibody. So the size we tried is from the 20 kilodalton to almost 100 kilodalton. So within this range, I think this is no problem. But we found as this protein size become bigger, the nanoparticle size will slightly become enlarged. So in this case, I suggest the user It's better to reduce this calcium phosphate concentration to reduce this particle size.
Okay. Good. So the next question is asking about the different cell types.
Yeah.
So have you tried to apply these telefactions into other primary cell lines, such as the primary haplocyte?
Okay, and it's a good question because I also study the liver. So in our lab we also prepare this primary hepatocyte culture. So I tried this telefaction delivery in the primary culture system, but this be honest, the efficiency is not so good for the plasmid delivery. It's maybe 1 to 2%. That is very low. So, but for this, we found for this protein delivery, it has very high transfection, high transfection delivery. It's maybe have this almost 90% protein delivery. This protein is an antibody, fluorescent conjugate antibody. So it have almost 90% protein delivery. Because we can detect this fluorescent signal degradation, and we found this protein can be uptake by this hepatocyte, primary culture hepatocyte, but they will be degrade within maybe 48 hours. The half life maybe 48 hours. So the problem, I think the problem is this cell can uptake the plasmid or protein, but this plasmid or protein will be degraded during then escape this endosome complex. So maybe one problem for this hepatocyte primary culture we need to do is to prevent this endocytosis DNA or protein degradation. This maybe will improve this transfection efficiency.
Yeah. I can see in the audience there are many people doing stem cell research.
Yeah.
I can see many questions asking, have you tried these methods on like embryonic stem cells and neuroprogenitor cells or induced pluripotent stem cells
Okay.
before
Yes.
differentiation? Yeah.
Yeah. Okay. So, okay, this slide I showed you before. Actually, we also try to use the telefaction to deliver protein and the plasmid into the neuron stem cell. We only try this neuron stem cell, and as you can see, this transfection efficiency show this positive correlation with calcium phosphate nanoparticle. It's when you increase this calcium phosphate nanoparticle number, this transfection cell number will increase. So we have this good transfection efficiency under this 200 microliter telefaction medium. So and I think for the neuron stem cell, it's no problem for the telefaction to deliver this gene plasmid and the protein, but we never try other kind of stem cell.
Yeah, I can see Sabrina is raising a hand in the audience. Could you please type your questions into the question and answer box, please? Yeah, the next question from Zhengping is asking on the other side of this process is can you apply these techniques with messenger RNA? Is it stable with telefactions?
Okay. We also try to use telefaction to deliver mRNA. mRNA can be delivered by telefaction, but in our case, maybe this we cannot handle this mRNA perfect. So it has, I think it has some degradation. So compared to this DNA delivery, mRNA delivery efficiency is, I think it's lower than this plasmid. Yeah, I think the major problem is the stability of mRNA, not for the telefaction itself.
Yeah. Okay. The next one is from Eric. So So he asked, it seems to have a low toxicity than other commercial kits. Does it have any side effect for the neurons during the telefactions procedures?
Okay.
Does the cells have any response or change during the telefactions
Okay.
transfection procedures?
Okay, so so this data we show in our supplementary data. So during this telefaction, we also try to uh, uh, monitor this cell physiology and some cell activity by this calcium image and the electrophysiology. So here you can see this the efficiency of a calcium phosphate nanoparticle. Sorry, the impact of this calcium phosphate nanoparticle on this electrophysiology of the primary neuron was shown as this line graph. Okay, so this is a kind of a sodium channel tracing with this IV curve. So the data indicate during telefaction, neurons show around 40% downregulation of sodium current, but it's still but this inhibition was rescued by replaced the telefaction medium with HBSS or conditional medium. And for the calcium image, we found this intracellular calcium was increased about 10% and showed it has a statistic significance. But this 10% calcium elevation did not impair NMDA mediated neuron activity. So actually, telefaction show only mild effects on this neuron physiology during nanoparticle incubation. So this can be reversed. This is reversible after releasing from this telefaction medium. So telefaction has an impact, but not so strong, and it is reversible.
Okay, great. We will take two more questions. Okay.
Yeah.
So Zhixiang's come back. He asked, is the calcium phosphate complex escaped from the endosomes via the low pH endosome lysosome transitions? And how would you think about the ratio of the population that successfully get to the cytosomes and then to the nucleus?
Yeah, I think this is good question, but we don't have answer right now, and we try to use different ratio and try to use different inhibitor or inducer to enhance this transfection efficiency, but it's still undergoing. So it's difficult for me to answer this question. Sorry.
So I think the last question just ask you to maybe highlight the specific advantage of using the calcium phosphate nanoparticles.
Mm-hmm.
Yeah. What's the specific advantage of using
more
this technique?
specific. Oh, okay. I think telefaction is very cheap and you can this is a homemade reagent. You can use very cheap calcium phosphate and prepare all the reagent in your lab. You don't need to buy this lipofetamine or other commercialized kits. And I think this for the neuron, it's a lower cytotoxicity compared to this control group. So I think this a good tool for especially for the primary neuron culture for the gene delivery. But people need to know. It's very critical information I show you here. You need to know. As you can see, this table shows the difference between this new neuron basal median and the conditional median. So this is very important when you use telefaction to transfer this plasmid into a primary neuron. We strongly suggest that you have to use this conditional neuron basal median. You cannot use the new neuron basal median because this the ionic balance, osmolarity, and the metabolites will be changed after several days of a cell culture. So this osmotic and ionic shock may be occur when switching from this conditioned medium to the fresh neuron basal medium in permeable neuron. Okay, because of change of these multiple subtle but important factor for the ion balance, and it may cause this cytotoxicity. So when you use this telefaction, please remember use this conditional neuron basal medium. Do not use the new one. But when you use telefaction, when you apply telefaction into this cell line system, that's no problem. You can use the new neuron basal media. Yeah. Okay.
Right. Thank you very much, Dr. Schumann Chao, for such insightful presentation today. And thank you to all the audience members for joining us for this webinars. There are more exciting Mechan Express webinars coming up very soon. Our next confirmed webinars will focus on the steroid receptor signalings in breast cancer. So please stay in tune for that. And at Mechan Express, we're committed to fostering the scientific communications and accelerating the pace of scientific discoveries. We thank you again for joining us today, and stay connected. And I look forward to seeing you all very soon. So goodbye for now.
Okay,
Thanks,
thank you for the invitation.
Bye.
Bye.